ATP in my system, in my cell, it turns out that ATP is going to provide negative feedback on the process of glycolysis in the Krebs cycle. Okay, that makes sense, doesn't it? But let's say that our ATP stores are low. How is the cell going to detect that? That it's low on ATP, it needs to make more ATP. What would be the chemical signal there? Any ideas? When your ATP levels are low, that means something else is high. What could that be?
Isn't it your insulin is high?
Here's the reaction back and forth, isn't it? If I have high ATP levels, my ADP levels are low, correct? If my ATP is low, then what's high? ADP. And so it turns out that ADP has a stimulatory effect on glycolysis. And both ATP and ADP affect the same enzyme in glycolysis. One stimulates, one inhibits. So ATP stimulates glycolysis. And it does that with the first or the second enzyme of the pathway. Very interesting levels of control in that way. We could also say that, of course, obviously, the availability of oxygen gas is an important variable here, isn't it? If I don't have O2, can I carry out aerobic respiration?
No.
No, because there's nothing to receive those electrons and hydrogens that are flowing. There's nowhere to go. There's nothing to receive them, so we cannot do aerobic respiration. So that's kind of assumed, but I'll leave that there. Now, the reason I left that up on the top, and we're going to come back to that, is that I want to link lipid metabolism and protein metabolism with carbohydrate metabolism. We're going to join them all together. And we also have that over here, but it's such a detailed map, and it's so beyond the scope of our course that you wouldn't want to study that. But it's a very cool device. So let's think about metabolism of lipids and proteins. We'll get started with that. We've primarily been talking about carbohydrate metabolism, although glycogen synthesis is anabolism, isn't it? So we did mention anabolism. So metabolism of lipids and proteins. We'll discuss this in less detail. And first we want to talk about lipolysis. With lipolysis, what's going to happen to lipids?
They're going to be split.
They're going to be split, aren't they? Exactly. So, lysis means splitting, and generally we do this with a hydrolysis reaction. So in lipolysis, we take a triglyceride lipid, and we break that down into glycerol plus 3 fatty acids. Glycerol plus 3 fatty acids. And these can be used to make ATP. Let's see how. So here's my lipid. I can make glycerol plus fatty acids. And what does the glycerol, what does the body do with the glycerol? The glycerol can go into glycolysis and enter as an intermediate of glycolysis and then travel down through this pathway. The fatty acids, through a process called beta oxidation, will produce a number of acetyl groups. One fatty acid that contains 16 carbons can make 8 acetyl groups. And what does the cell do with those acetyl groups? We can make a ton of energy, can't we? We skip glycolysis, the minimum wage pathway, right? And we jump to the high wage pathway and get lots of ATP. So that's what we get with this process. So with, what have we done now? We've linked lipid metabolism into carbohydrate metabolism, haven't we? They have common pathways, don't they? They really do. Okay. Now, what happens in people who either have diabetes mellitus, sugar diabetes, or in people who are on the Atkins diet, or say the people that were in the Donner Party when they were starving and they were living off their fat reserves, and then eventually they lived off their protein reserves. I mentioned the Donner Party today because I had a day and a half power outage last weekend, and I thought it was appropriate to read about the Donner Party, so I went back to I read Winter of Entrapment. It's a good book, and kind of enjoyed that. Anyway, so because I thought, well, I know they're a lot worse off than I was. I had my wood stove going, you know, I had no water and no electricity, but I knew that was happening. And so, anyway, let's talk about what happens in these people, like the Donner Party folks for a while they were alive, in fasting. So you're not getting any carbohydrate input into your diet. Your body is trying to make some sugars from the fat that you're digesting. And you're digesting a lot of fat like crazy. So in this fasting, what happens? We have lots of the beta oxidation. We have excess beta oxidation. And this results in the accumulation of ketone bodies.
Can you tell me why you call it beta oxidation?
Sure. Sure can. Beta is the second letter of the Greek alphabet. And if we draw a fatty acid, we're looking at this molecule. I haven't fleshed out all the other elements on the fatty acid tail, but when this attack comes, it attacks this bond right here. This is the so-called beta carbon. Alpha, beta. In other words, the first and second. So we attack that carbon, and what do we attack it with? Coenzyme A. And what do we have? Acetyl coenzyme A. So we get the 2-carbon unit attached to the coenzyme A, and we do another one, and so forth, all the way down the line. So that's the beta. The number 2 carbon. All right. Beta oxidation produces acetyl groups. So we get the accumulation of these ketone bodies. How do we detect these ketone bodies? We can smell a fruity breath on these people, can't we? They perspire, we can smell that. The urine has an odor to it. We can detect ketones in the urine with a dipstick. Okay? And so what does this mean? Well, we get acetoacetic acid accumulating. So some of the ketone bodies are acids, and these people then may suffer from ketoacidosis. Okay? This can lead to ketoacidosis. So your body pH is dropping from the increased keto acids. Why are we getting these accumulated? Because you're pouring so much into this spot that the pickup molecule, oxaloacetic acid, becomes overwhelmed.